Welcome back to Acing AP Biology. In this video, we will begin our discussion of the cell. The cell is the fundamental unit of life. All cells have a plasma membrane, cytoplasm, which is made from cytosol, which is the watery substance inside the cell, and genetic material, which could be DNA or RNA. Cell theory states that all life is composed of one or more cells. Cells are the fundamental unit of life, and all cells arise from pre-existing cells. The size of a cell must remain in a certain range, as is required for cellular activities. The cell must be large enough to house all the organelles and DNA, which is held in the nucleus. However, the cell cannot grow too large either. When cell size increases, the volume increases much more proportionately than the surface area. The cell must maintain a large surface area to volume ratio, because a large surface area provides the cell with enough surface for substance exchange with its surroundings. For instance, the surface area must be large enough for sufficient exchange of oxygen, nutrients, and waste products with its surroundings. In addition, if cell size grows too large, it may become too big for the nucleus to control, and it won't be able to oversee all the cell's functions. For this reason, larger cells sometimes have multiple nuclei. Also, as cell volume increases, transport becomes slower and may become too slow for cell function. Thus, a moderate size for a cell is key for its function. Now let's talk about the difference between a prokaryotic and a eukaryotic cell. A prokaryotic cell is only found in bacteria and archaea. They do not have a true nucleus, nor do they have a nuclear envelope, which is the membrane surrounding the nucleus. So, their genetic material is held in a region called the nucleoid. They also do not have membrane-bound organelles. Eukaryotic cells are found in plants, animals, protists, and fungi. They have a true nucleus surrounded by a nuclear envelope. Their genetic material is found in the nucleus. And lastly, they have membrane-bound organelles. The internal membranes are very important as they sequester the biochemical processes going on within each organelle from the rest of the cell. This allows for each organelle to have a unique environment specific to its function. Let's look at a diagram of the prokaryotic cell. As you can see, it has a plasma membrane, cytoplasm, and genetic material held in the nucleoid region. The genetic material is organized as a single circular chromosome. Surrounding the plasma membrane is the cell wall, which adds extra protection. Some prokaryotes have a capsule outside the cell wall, which is made of sugar and is used for attachment. The fimbriae, which are hair-like structures, are also used for attachment. Ribosomes are organelles used for protein production. And finally, flagella help the prokaryote with locomotion. Now let's take a look at the animal cell. The cell is surrounded by a plasma membrane composed of a phospholipid bilayer. Notice that there is no cell wall in an animal cell. Inside the cell, there is cytoplasm, made up of cytosol. The nucleus contains the genetic material, DNA in this case, that provides instructions for cellular activity. The nucleus is known as the control center of the cell. DNA is organized as chromatin. Condensed chromatin forms chromosomes. The nucleolus produces ribosomes. The nuclear envelope is a double membrane around the nucleus. The rough endoplasmic reticulum gets its name because it has a rough appearance due to the presence of ribosomes, which are protein factories. The rough endoplasmic reticulum produces secretory proteins and membrane proteins. The smooth endoplasmic reticulum produces lipids and detoxifies toxins. The Golgi apparatus modifies, sorts, and labels proteins coming from the rough endoplasmic reticulum. A vesicle pinches off the Golgi and transports the protein to the plasma membrane or other areas of the cell. The lysosome, which is unique to the animal cell, digests macromolecules and breaks down waste material. The mitochondria is known as the powerhouse of the cell, producing energy in the form of ATP for the cell to use. The peroxisome, which only has a single membrane rather than a double membrane, detoxifies alcohol and other toxins. They break down hydrogen peroxide into water. The cytoskeleton provides shape and support and allows for movement. The cytoskeleton is composed of microfilaments, intermediate filaments, and microtubules. The centrosome is the organizing center for microtubules. The centrosome contains centrioles, which are unique to animal cells. Some animal cells have microvilli to increase surface area. And finally, some animal cells have flagellum made of microtubules that are used for motility. The animal and plant cell share a lot of similarities. They both have a plasma membrane, cytoplasm, a nucleus, ribosomes, the rough ER, the smooth ER, the Golgi apparatus, vesicles, mitochondria, cytoskeleton, and peroxisomes. However, plant cells have organelles that are unique to plant cells, such as the cell wall, the central vacuole, the chloroplast, and plasmodesmata. The cell wall is made up of cellulose, the structural polysaccharide we talked about in Unit 3. The cell wall provides protection, support, and strength. There can be a primary cell wall and a secondary cell wall, which we will discuss in more detail later. The central vacuole stores water and secondary compounds like pigments, and it exerts pressure. The membrane surrounding the central vacuole is called the tonoplast. Then we have the chloroplast, which is responsible for photosynthesis, converting light energy to chemical energy. And lastly, plasmodesmata are used for material transport between plant cells. In the next video, we will explore the functions of each organelle in detail. See you then. For more bio content, hit subscribe.